One of the big problems in the field of neurodegenerative disease, particularly Alzheimer's disease, is that it's very difficult for us to predict when we make discoveries in the laboratory which things are going to make a difference for patients. And I think it's a frustrating issue for patients who often read about mice getting cured for Alzheimer's disease in the news, and then they don't see the cure in the clinic. And I think we were really struggling with why is that the case? And we wanted to really look at that interface between what we do in the laboratory and what we do with patients in the clinic. And so that's a really difficult problem to study, though, because the two groups who normally work on those two issues work in separate worlds almost. And I think in this case, we had the unique opportunity to bring together two groups, one at the Memory and Aging Center at UCSF and a group at the Gladstone Institutes that works on Alzheimer's disease to study how are mice and humans different or the same as we study Alzheimer's disease.
So what we try to do is actually facilitate the translation of these discoveries by making the methods to test memory very similar in animal models and in patients affected by the early stage of Alzheimer's disease.
The Morris maze is a measure of spatial learning and memory. In Alzheimer's disease, patients often have impairments in spatial learning and memory early in the illness. In fact, some even get lost. And the reason for this is that this skill relies on the hippocampus, a brain region that is hit early by the disease. And so the Morris maze is a really good choice to measure early impairments of Alzheimer's disease. So we designed an analogous version of this test that is like a video game, similar to how you might find your way across the city, learning where your office is located relative to other major landmarks in the city. The mice and the humans have several trials to learn to find a target. The target is always hidden in the same place, and so both the mice and the humans learn to find it by learning its relationship to cues, these landmarks that surround where they're navigating. And what we found was there's really a dramatic difference where the mouse model of Alzheimer's disease and also the patients who have a very early stage of Alzheimer's disease don't learn as well where that target is located, and they also don't remember as well where that target was located after the delay.
Many of the measures that we selected, and even the one that we developed for this study, we found that the effect size of the deficits were extremely similar.
The most common method for analyzing Morris-Water maze data is called the repeated measures ANOVA, but unfortunately this method is not the most appropriate method. We present instead a simple rank summary score method that is appropriate to apply statistically and is actually very easy to apply. We show that it's at least as sensitive as more complex methods.
So one important recommendation that we provide in this manuscript is just a number of subjects that will have to be enrolled in a study either for the animal research or the human clinical studies to get enough power to detect not only a deficit but to also detect a potential effect of a drug. So we think our study proved the concept that if you bring clinicians and basic scientists together, you can actually build a translational platform in which you're using methods that are very similar to test for functional deficits. In our study, it's spatial memory, but you could imagine that you could apply that for different purpose or different disease.
This study, I'm very excited about it because it's really the first that I can think about that explores this specific interface between what we do when we study mouse models of these diseases and what we're doing with patients.